The Subcommittee on Research and Technology will come to order. Without objection, the gentleman from Texas, Mr. Weber, is authorized to participate in today's hearing. And without objection, the chair is authorized to declare recesses of the subcommittee at any time. Good morning and welcome to today's hearing entitled The Future of Biotechnology, Solutions for Energy, Agriculture, and Manufacturing. In front of you are packets containing the written testimony, biographies, and truth and testimony disclosures for today's witnesses. I now recognize myself for 5 minutes for an opening statement. Humans have used biotechnology since the dawn of civilization, manipulating biology to improve plants and animals through hybridization and other methods. Rapid advancements in scientific knowledge and technology throughout the 20th century gave rise to the field of modern biotechnology, making useful products to meet human needs and demands. Biotechnology has become part of our everyday lives, from producing the insulin used by diabetics to the corn we eat and use to produce fuel to the detergent that cleans our clothes. Today we are here to discuss what the future of biotechnology will look like in this century, specifically for solving some of our greatest 21st century challenges in energy, agriculture, and manufacturing. In June, the subcommittee held a hearing on the science and ethics of genetically engineered human DNA. The hearing looked at the research and issues surrounding the application of new gene editing technologies for human health. I hope that today's hearing will build upon that fascinating discussion and help inform a research and regulatory framework that continues to ensure safety without stifling innovation. The biotechnology and biological science industry is a sizable and growing economic driver in our country. In Virginia, the industry employs over 26,000 people across 1,500 companies and institutions, including the George Washington University Ashburn Campus Computational Biology Institute located in my district. Here they apply technology tools to a variety of funded research in pediatric medicine, coronary heart disease, cancer, Alzheimer's disease, and schizophrenia, just to name a few. These are good-paying jobs, and I want to find ways to keep these jobs in the United States and encourage young people to study the STEM subjects needed to fill these jobs and create new ones. But more importantly, these are jobs and an industry that's going to improve our way of life and improve our health and save lives. So I appreciate and look forward to learning more about these new and emerging technologies and their applications, understand better the role of the federal government in funding and regulating biotechnology, and hear from the witnesses about the economic benefits to the United States and how we can stay on the cutting edge of innovation. Now, I now recognize, I guess our ranking member is not with us yet this morning, but will be joining us shortly. I know he just had a little bit of a flight delay, but will be with us shortly, and I appreciate Mr. Lipinski joining us, and we will recognize him at that time. But let me see, we will, if there are members who wish to submit additional opening statements, your statements will be added to the record at this point. Now, at this time, I would like to introduce our witnesses. Dr. Mary Maxson is the Biosciences Principal Deputy at Lawrence Berkeley National Laboratory. She has previous experience as Assistant Director for Biological Research at the White House Office of Science and Technology Policy, or OSTP, and has worked for a variety of biotech organizations. Dr. Maxson earned her PhD in molecular cell biology from the University of California, Berkeley, and did postdoctoral research in biochemistry and genetics at the University of California, San Francisco. Our second witness today is Dr. Steve Evans. Dr. Evans is a fellow for advanced technology development at Dow AgroSciences. Dr. Evans is the past chair of the Industrial Advisory Board of the Sinbirk Synthetic Biology Consortium, funded by the National Science Foundation. Dr. Evans earned his bachelor's degrees in chemistry and microbiology from the University of Mississippi and his PhD in microphysiology from the University of Mississippi Medical Center. Today's third witness, I got my glasses here. Today's third witness is Dr. Reshma Shetty, co-founder of Ginkgo Bioworks. Dr. Shetty served as an advisor to the International Genetically Engineered Machines competition, where she was best known for engineering bacteria to smell their bananas to smell like bananas and mint, and was named by Forbes as one of the 8 people inventing the future in 2008. Dr. Shetty earned her bachelor's in computer science from the University of Utah and a PhD in biological engineering from MIT. Testifying next is Dr. Martin Dickman, Distinguished Professor and Director for the Institute for Plant Genomics and Biotechnology at Texas A&M. Dr. Dickman's research focuses on the genetics and molecular biology of fungal-plant interactions, and he established that parallels exist between plant and animal systems, disease and infection strategies. Dr. Dickman earned his PhD from the University of Hawaii. Our final witness is Dr. Zach Serber, co-founder and vice president of development for Zymergen. Dr. Serber previously worked as director of biology at Amyris and as a research fellow at Stanford University Medical School. Dr. Serber earned his bachelor's degree from Columbia University, his master's in neuroscience from the University of Edinburgh, and his PhD in biophysics from the University of California, San Francisco. As always, we are so honored to have such distinguished and accomplished witnesses joining us here today. And I now will recognize Dr. Maxson for 5 minutes to present her testimony.
Chairwoman Comstock, members of the committee, thank you for holding this very important meeting and for inviting me to participate. I applaud the committee for exploring the great potential that advanced biology has to address the nation's grand challenges and to stimulate innovation. I believe a federally coordinated strategic program that leverages the national labs and other existing federal capabilities would greatly accelerate this. I'm the Biosciences Principal Deputy at Lawrence Berkeley National Lab and have enjoyed a 30-year career as a biologist. Recently, I served as Assistant Director for Biological Research at the Office of Science and Technology Policy, where I was the principal author of the National Bioeconomy Blueprint. Although my testimony represents my own views, I would be remiss not to recognize the leadership of the Department of Energy and Berkeley Lab in driving the nation's engineering biology capabilities forward. In particular, DOE's Office of Biological and Environmental Research supports some of the nation's most foundational resources in this field. DNA can be viewed as a programming language, where instead of the ones and zeros that are used to program computers, A's and C's and G's and T's, the building blocks of DNA, are used to program biology for useful purposes. Biology can improve agricultural yields, increase nutrients in soil, reduce the need for water and fertilizers. It can be used to create biosolutions to reduce the demand for livestock-based protein sources, such as beef and poultry, and for a planet with more people and fewer resources. It can convert non-food biomass into fuel and chemicals, and in the process, replace fossil fuels. It can convert microbes into low-cost producers of drugs and alter microbiomes to improve human and animal health. Although DNA sequencing, that is reading DNA, thanks in large part to the Human Genome Project, is fast, cheap, and democratic, meaning that researchers everywhere can now sequence DNA themselves. Engineering biology, that is writing DNA, remains slow and expensive. National labs can help change this dynamic. They can play important roles in harnessing biology to meet national scale challenges, and in doing so, democratize engineering biology to enable researchers everywhere to drive advancements across fields of science and industrial applications. But currently missing from this collection of currently missing from the collection of national laboratory user facilities is a biofoundry, a high-throughput, open engineering biology facility powered by capabilities in physical sciences and supercomputing to develop freely available tools, technologies, and knowledge needed to accelerate engineering biology and drive a sustainable national bioeconomy. Such a facility could accelerate scientific discovery, reduce costs, and time to market for new bioproducts that are needed to transform manufacturing processes for both human and environmental benefit. It would build on and capture a greater return on DOE's existing investments in genome sequencing, synthetic biology, and other engineering research capabilities. Berkeley Lab has made an initial investment to launch an open biofoundry, and has undertaken early proof of concept work aimed at establishing a robust democratic platform technology for the engineering of biology to provide fundamental advances needed to transform manufacturing to reduce energy intensity and negative environmental impacts of traditional manufacturing. Recent industry listening sessions held by Berkeley Lab indicate that in addition to user facilities, national labs can serve at least 4 unique and important functions for industry. 1, meet vital research needs that are considered off mission by the company investors. 2, validate technologies from the academic sector for companies, which is currently a time-consuming and frequently unproductive endeavor for industry, and provide for the transfer of technical expertise and capacity building directly by embedding industry researchers in the biofoundry. And lastly, by providing access to flexible pilot-scale production facilities to enable research advances in understanding how to predict large-scale production of bioproducts, currently something of a holy grail. I applaud the committee for its interest in the topic of engineering biology and believe that a vision for a strong, long-term research and development program, including research in the ethical, environmental, and social aspects of engineering biology, is needed for the U.S. to lay a solid foundation on which to build a robust and responsible biomanufacturing future, create new markets and jobs, and drive the U.S. bioeconomy. Thank you.
Thank you, Doctor. And Mr. Lipinski has now joined us, so I'm just going to take a little break here on the witness testimony and allow Congressman Lipinski to give his opening statement.
Thank you, Chairman Comstock, and thank the witnesses for being willing to deal with this little interruption here. I want to thank the Chairman for holding this hearing and look forward to, I thank Dr. Maxson for her testimony, look forward to all the testimony here this morning. One of the reasons I chose to be on the Science Committee, in the subcommittee in particular, is that we have the opportunity to learn firsthand about new and emerging research fields and technologies that will transform society, and to hear what the federal government can do to help society benefit from these technologies. This morning is no different. Today we'll hear about new technologies that have the potential to transform the energy, agricultural, and manufacturing sectors. A number of these new biotechnologies are based in engineering biology research, which is research at the intersection of biology, physical sciences, engineering, and information technology. This emerging field has been fueled by the development and increased affordability of technologies such as DNA sequencing and DNA synthesis. In the case of DNA sequencing, the Human Genome Project, an international research project to sequence the human genome, was coordinated by the Department of Energy and the National Institutes of Health. And it took over a decade and cost $2.7 billion. Remarkably, sequencing the human genome now costs less than $1,500. Several agencies under this committee's jurisdiction have significant programs in engineering biology. The Department of Energy has invested in programs focused on bioenergy. The National Science Foundation has invested in this area both in individual research awards and through their support of an engineering research center, SINBERG, at UC Berkeley. NASA and NIST also have programs in this area. NIST has a particularly important role in the development of technical standards for a future biomanufacturing economy. And of course, agencies outside the committee's jurisdiction, including DARPA, NIH, and the Department of Agriculture, are also significant players in this research. Due to the importance of this growing research field, the nation would benefit not just from increased investment at individual agencies, but also from coordination of federal efforts under some kind of national plan or strategy. Additionally, we should ensure that we are facilitating public-private partnerships. Given the potential commercial applications across nearly all sectors of our economy, there is a need to engage and encourage private sector collaboration at a pre-competitive level. I look forward to hearing from all of our private sector witnesses what they're looking for in partnerships with federal agencies, national labs, and universities. And finally, we must pay careful attention to the issues of human and environmental safety and ethics when it comes to engineering biology research, including support of research on these topics. The future of biotechnology could include automotive and even jet fuels produced cheaply, cleanly, and safely by specifically engineered bacteria. Also, more drought and pest-tolerant crops and feedstocks, and also transformation of materials and manufacturing with applications across our economy. These technologies would have significant economic benefit for the United States. So it's important that we make the necessary federal investments in the foundational research and partner with the private sector across the potential application areas. Look forward to the rest of the witness testimony and the Q&A, and I thank you for being here today. I yield back the balance of my time.
Thank you, and I will now recognize Dr. Evans for his 5-minute testimony.
Good morning, Chairwoman Comstock, Ranking Member Lipinski, and the members of the House Subcommittee on Research and Technology. Thank you for inviting me here to represent my company, Dow AgroSciences, in this hearing on emerging biotechnology applications. We trace our roots in agriculture back 60 years, and we emerged from the Dow Chemical Company, a company that's been transforming technology into viable solutions since 1897. The drivers for application of biotechnology into agriculture are clear. The global demands for food, fuel, fiber, and feed are strong and rising. The solutions to meet this global need must be met within increasing constraints and unpredictability, reinforcing the need to make newer product offerings even more sustainable. We have all heard of the challenge set forth for global needs by 2050, and between now and that point in time, agriculture will need to produce more food than the sum total of what has been produced in the last 10,000 years. Since their introduction in the mid-1990s, agricultural biotechnology offerings have made significant contributions to global food security, and biotechnology-based crops are the fastest adopted crop technology in the history of modern agriculture. If you were to visit an early-stage laboratory in R&D laboratory in Dow AgroSciences, you would see the tools and techniques that are used in common bioscience endeavors. Early-stage ag biotechnology benefits from the same molecular biology, bioinformatics, DNA sequencing, high-throughput analytical systems, and other advances from basic life sciences that have been funded by federal research. One of the ways that Dow AgroSciences has benefited from advances in related fields is by being able to provide input and shape ideas for technology in something like the NSF Engineering Research Centers. SINBRC, as has already been mentioned, brings together 37 professors, 18 universities, and 47 companies with the stated mission of making biology easier to engineer. As past chair of that industrial advisory board, I note that a portion of the companies there represent established ag companies, but also smaller startups with concepts in the agricultural space. The ERC provides a unique pre-competitive venue for industry participation and influence in the technology development. And some of the tools that have been developed there are now being brought into our company directly and used by Dow AgroSciences. I recently examined some patent activity by other ag players, and you can see that those technologies are being broadly adopted at the early stages of most of the agricultural companies. But to really understand and develop a realistic expectation for when these things would appear in agricultural products, you have to understand a little bit about biotechnology development timelines. The typical range of development spans 7 to 10 years and an average investment price tag of over $130 million per product. While laboratory tools and technologies just described play an important role in performing and accelerating that front end, we're still faced with multiple challenges at national and international regulatory frameworks. Companies understand and can manage the risks related to product performance and customer choice. However, because of the time horizon of nearly a decade and a cost of $100 million, to make informed investment decisions, we need to have a regulatory approval process that is predictable to enable scientific planning. That regulatory process needs to be science-based and proportionate to risk. In addition to using biotechnology for modern crops, we have an offering in Dow AgroSciences that is based on agrochemicals derived from natural products. We, taken together, products and chemistries that are inspired by natural products account for 1/4 of the global ag chemistry sales. One challenge in developing those natural products, whether for pharma or ag, is that we need to attain sufficient productivity to make that product economically viable. Dow AgroSciences' platform to integrate those biotechnology tools, either from external sources or from internal capabilities aimed at rational engineering of our strains, is how we use engineering biology in our platform. Nationally funded research has enabled key milestones in that field, but the U.S. is not alone in recognizing the economic and environmental benefit to be derived from commercial manufacturing of novel natural products or chemistries inspired by them. So finally, I will propose that a framework for involvement of the federal government can be understood in terms of 3 C's. Number 1, continue to support exceptional science. Number 2, convene forums for discussion on development and risk proportionate oversight. And number 3, create a strategic vision for the U.S. biotechnology investments to produce exceptional solutions for the world's most pressing needs. These actions are important to maintain a U.S. position of leadership and development in this technology, and it's an increasingly competitive and global race. Within these fields, these investments provide technology, a workforce of new skilled talent, and predictable science-based regulatory framework from which companies like ours can make informed investment decisions for products taking over a decade to bring to market. Thank you.
Thank you, Dr. Evans. Now I'll recognize Dr. Shetty for a 5-minute statement.
Chairwoman Comstock, Ranking Member Lipinski, and distinguished members of the subcommittee, I would like to thank you for the opportunity to testify here today on the future of biotechnology and its applications in energy, agriculture, and manufacturing. My name is Reshma Shetty, and I'm a co-founder and president of Ginkgo Bioworks, a biotechnology startup in Boston, Massachusetts. I hold a PhD in biological engineering from MIT and have been active in the field of biological engineering for over 10 years. Today, I was asked to testify a little bit about Ginkgo's story as a case study for how federal investment in emerging technologies can stimulate the growth of new companies and new industries and make recommendations for how the U.S. government can continue to stimulate the growth of the domestic biotechnology industry. Ginkgo is an organism company. We design and build microbes such as yeast to spec for customers. Our customers use Ginkgo microbes in fermentation. Fermentation is a process by which cooking is done with microbes rather than heat. Humans have been fermenting foods and beverages like yogurt, beer, and wine for more than 9,000 years, so it's a very old technology. At Ginkgo, we design yeast to make new products from fermentation, or what we call cultured products. Our first commercial organisms are microbes for the production of cultured ingredients, so ingredients that end up in household consumer goods, things like sweeteners, flavors, fragrances, vitamins. So for example, Ginkgo is developing a yeast to produce a rose fragrance, what we call a cultured rose. Other companies are making cultured products such as animal-free cultured leather, animal-free cultured meat, cultured milk, cultured silk for making jackets, and so on. I started Ginkgo in 2008 with 4 fellow MIT PhDs, including Tom Knight, who's widely considered to be a father of the field of synthetic biology. Quite frankly, at the time, I knew almost nothing about what it took to start and run a company. What I did know was that biological technologies were going to be incredibly important in this century, and I had ideas about what were the important technologies to be working on and developing. Federal grants and contracts from the NSF SBIR program, DOE ARPA-E, NIST, and DARPA all provided absolutely critical funding for Ginkgo in our early days as we transitioned from MIT and university life to the real world. Today, we've raised more than $50 million of private investment, have built an 18,000 square foot facility in Boston for manufacturing of microbes, and we have commercial contracts for more than 20 different cultured ingredients. In In the last 6 months, we've doubled our workforce, and more than a quarter of which actually live in the 5th District of Massachusetts and are represented by Congresswoman Clark. In short, your investments helped make Ginkgo what it is today. In the early days of the computer industry, the U.S. government played a critical role in nurturing the nascent industry through both R&D funding and through serving as an early customer for integrated circuits via the Apollo program. This federal investment was critical in creating demand for integrated circuits and stimulated a significant later private investment in the space. The computer industry would not be the major economic and job engine for the U.S. economy that it is today if it weren't for the U.S. government's role. I believe that the U.S. government has an opportunity to play a similar role in the emerging biological engineering industry. Ginkgo itself is evidence of the payoff that federal R&D investments can generate, and I urge you to build on these early R&D investments in this space by recognizing the importance of the U.S. government as an early adopter of biotechnology products. With countries like the United Kingdom and China having well-coordinated national programs in this area, the United States is at risk for losing its competitive edge. By serving as an early customer and stimulating demand for the products of biotechnology, the U.S. government could play a central role to biotechnology today as it did in the 1960s to the computer industry. In short, I suggest that the committee, 1, enhance U.S. competitiveness in biotechnology via direct R&D funding for public domain foundational technologies that are available for all to use without intellectual property restrictions. 2, continue to garner bipartisan support for H.R. 591 to establish a national engineering biology research and development program. And 3, recognize the importance of the U.S. government as an early customer for biologically engineered products. Thank you.
Thank you. And I now recognize Dr. Dickman for 5 minutes.
Thank you, and good morning, Chairwoman Comstock, Ranking Member Lipinski, and members of the subcommittee. Thank you. I am, among other things, I am also the director of the Norman Borlaug Center at Texas A&M University, and my background is in, actually, I'm a plant pathologist specializing in fungal diseases. But I wanted to use that title as a prelude to just mention Dr. Borlaug, who is largely responsible for the development and implementation of the Green Revolution. And he has been widely acclaimed for this work, including such awards as the Nobel Prize, which is the only agriculturist to be awarded this honor, the U.S. Presidential Medal of Freedom. He's in company with Mother Teresa, U.S. Congressional Gold Medal, and on and on and on. But what Dr. Borlaug represented, besides breeding plants that had desirable attributes, was a dogged determination to try to ensure as best possible of feeding people throughout the world. And he's had a modicum of success with that. In fact, he is one of his other achievements is that he saved a lot of lives, but he is considered to have saved more lives than any other living human being ever. So the mission of our institute, the Institute for Plant Genomics and Biotechnology in the Borlaug Center, is to foster these ideals and progress using what's available and these developing technologies that we've heard a little bit about already this morning to increase our understanding of how things work in the ag biotechnology space. We want to improve agronomic traits for crop plants, and importantly, we want to prepare young scientists with the necessary technical and conceptual tools to face the inevitable challenges that lie ahead. As food safety and food security concerns continue and are likely to increase, it is clear that a new green revolution is needed. There is increased urbanization, limiting land availability, increased water use and energy demands, unpredictable climate changes, coupled with pollution and soil erosion. When taken together, collectively, they all contribute to a reduction in yield. And from a grower's point of view, yield is certainly the bottom line. We now face the task of growing more food on the same or even diminishing amounts of land. So in the remainder of my time this morning, I just want to highlight 3 biotechnological approaches that have varying degrees of risk, but all have the potential for really, really high rewards. And again, because of time, I will pick and choose some of the success stories that we in the Institute, as well as around the world, have employed to address some of these biotechnological approaches. So the three approaches I'm going to talk about are synthetic biology, which has already been mentioned, the phytobiome, and of course genome editing, which you've heard about in the past, and their impact on agriculture and food production. So in terms of synthetic biology, I'm going to talk about a cotton project that we have been undertaking in Texas A&M. Cotton has a very, very high degree of protein in its seed. It's about 25%, that's a lot. Therefore, the potential of cottonseed to help feed people is evident. However, the cottonseed also contain immune problems and cause male sterility, thus sort of precluding their application in the real world. So breeders at A&M very simply bred out that particular compound called gossypol, so it was no longer present in the plant and everything looked pretty good. Problem was, gossypol is also a defense compound in plants, limiting insect and fungal diseases. And when you got rid of gossypol, the plants were basically open game to these pathogens and parasites. So the operation was a success, but the patient died. So how to explore this was done with some of these new techniques, which I won't get into too much detail unless we're interested. And that is using virus-induced gene silencing and genomics and synthetic biology. A worker at A&M was able to not only knock the gene out that made gossypol, but also direct that construct into the seed tissue itself. These are very nice, powerful, significant techniques. So now gossypol would be expressed in the plant. However, it would not be expressed only in the seed. Therefore, they were gossypol-free, and the seed could be produced, okay? To give you an idea of the scope of this, and we have several patents filed and many, many field tests that are going on around the world with these cotton plants, is that it is estimated that with the addition of this cottonseed as a protein source, 500 million more people can now be fed. So this is the kind of conclusions we'd like to see more of and highlight, but it also illustrates the approaches. There was no way these experiments and these conclusions could have been obtained without biotechnological approaches that were implemented and have been relatively new on the scene. Now, the other sort of crop example I want to give is bananas. Oh, wow, I'm going to hurry up. Bananas, very quickly, are seedless, and I'm not going to go into the details, but if you go to the store and buy a banana, there's no seed. Therefore, genetics and breeding are impossible. Now, bananas are a staple in a number of developing countries, and when they have diseases now, there's no program to study these diseases and solve this problem. So we have transgenic approaches going on with bananas right now, both in Africa and Australia and in the US, that are successfully impacting banana diseases. If banana diseases are uncontrolled in banana-consuming countries, people starve, people die. All right, the next topic I'm going to whiz through is the phytobiome. And all I want to point out here is it turns out that the microflora, the endophytes, if you will, is a fancy word, and it found in virtually all plants impact a great deal of attributes that enhance the crop in question. So the phytobiome, for example, will enhance drought tolerance, disease control, but only in the areas where they need this to happen. So in work, for example, done by Dr. Rodriguez, he found in Yellowstone that there's certain type of microflora associated with thermal tolerant plants, but differed from microbiome in the ocean, which conferred salt tolerance. So what I'm getting to is the fact that we can utilize phytobiome research to establish these probiotic microorganisms, and plants will make the necessary changes to control the stress that they are faced with. This is a new, high-risk, but very user-friendly control mechanism. The last part is involving CRISPR, which I know you've already heard about. So all I'm going to say about CRISPR, which doesn't have the same implications as the human application, CRISPR in plants has two major advances that are very exciting to the plant community. One is multiplexing and the ability to put numerous gene mutations in one genetic background. And the other is breeding. CRISPR is likely to revolutionize breeding. Breeding is a game of creating variation in plants and then going through all the characterization that needs to be done to understand the nature of that variation. Well, with CRISPR, you can make unlimited genetic variation by using this tool, thus obviating the need for chemicals and all the considerable work and time and effort that need to be done. So I will just come to my conclusions and just say that I want to remind everyone that all food that's consumed is genetically modified. We need a new green revolution to face the coming needs and continuing needs of people throughout the world. And we need to support basic research to make the conclusions verified. In other words, many of the great discoveries were only done by unintended consequences. Penicillin being a good example, which was, you could call him sloppy microbiologist. He found contamination that was inhibiting bacteria, and really that's the key. And the other one is CRISPR, which is really a study of immune issues in a bacterium, led to the CRISPR technology that we've discussed in quite a bit of detail here and in previous meetings of this group. Thank you.
Great. Thank you, Dr. Dickman. And I know we did go over our time, but you're educating us, and so I figured it's better for you to take up that time probably than some of us. So thank you, and we appreciate your enthusiasm, all of you. And now recognize Dr. Serber.
Good morning. Thank you, Chairwoman Comstock, Ranking Member Lipinski, and the rest of the committee for the opportunity to testify today on a topic that I've devoted my career to: expanding the impact of advanced biotechnology. A decade ago, I was one of hundreds, if not thousands, of early career scientists and engineers who left academia to devote our human capital to extending the reach of biotechnology. Whereas biotechnology is synonymous for many people with the field dedicated to medical therapeutics, biotechnology also has the potential to transform other fields, including energy, agriculture, and manufacturing. The prospect of vastly expanding the societal and economic impact of our technical expertise attracted me and many other scientists, including Dr. Shetty, to new endeavors focused on realizing these potential far-reaching applications. Single-celled organisms, microbes, are the most versatile chemical factories on the planet. Dr. Shetty has already explained how engineering microbes can be used as microscopic biofactories. This is the basis for what has been dubbed the new bioeconomy, in which companies increasingly rely on biology to source the materials used in their products. This is, however, not a new manufacturing paradigm. Today, chemicals made via large-scale fermentation are employed in a wide variety of agricultural and industrial applications, and, excluding ethanol, comprise over $66 billion in revenue globally, or roughly 10% as much as petrochemicals. While a relatively small percentage, the rate of growth of chemicals made biologically is greater than 10% annually, whereas the petrochemical market is growing at less than 7%. In time, chemicals made via fermentation may come to dominate the overall chemicals market. My company, Zymergen, was founded recently in 2013 to contribute to this expanding market. Our core business is to use biotechnology to rapidly and reliably engineer microbes used in the manufacturing of chemicals for a variety of applications. Zymergen is under contract with Fortune 500 companies to improve the manufacturing economics of chemicals they currently make in large-scale fermentation by engineering the single-celled biofactories they already use. Our ability to realize this incredible potential relies not only on scientists and engineers, but also on government policy that supports this type of research and innovation. Having interacted with dozens of large domestic producers of goods made through fermentation, I should mention that Zymergen fully supports the July 2nd White House memorandum on modernizing the regulatory system for biotechnology products, which directs the relevant federal agencies to develop a long-term strategy to ensure that the biotechnological regulatory system is prepared for the rapidly changing future of our industry. I can confidently say that the current regulatory system is full of inconsistencies and scientifically unsound characterizations. This regulatory system has not kept up with changes in the technology, creating confusion, delays, and inefficiencies. It is our hope that the EPA, FDA, and USDA can efficiently and rapidly update the coordinated framework. 2.5 years ago, Zymergen had 3 founders. Today, we have 93 employees. Growth has not slowed, and we are on pace to more than double in staff size in 2016. This rapid growth is not based on speculation. Quite the contrary. Our challenge to date has been excessive market demand. We are working day and night to keep up. Our customers are large, established manufacturers of chemicals made through fermentation. As they seek to reduce costs and increase manufacturing productivity and competitiveness, they see Zymergen and our technology as essential to maintaining competitiveness. Zymergen depends on cross-disciplinary research. Our engineers and scientists are trained in fields including microbiology, genetic engineering, robotics, chemical engineering, and machine learning. Our most valuable employees are rare individuals with expertise in multiple relevant domains, able to bridge the gaps between, for example, genome editing and software engineering. Federally supported educational and training programs are critical to providing us with the staff we need to grow and fulfill our potential. Recent activities in our space, supported both through public and private sector investment, have dramatically altered what is now possible through biotechnology. So while Zymergen has initially devoted our insights to improving the economics of existing products, the approaches developed enable us also to expand the palette of chemicals that can be made through biology. This amounts to a technological revolution likely as important to advancing societal well-being, national security, and economic productivity and competitiveness as the invention of the transistor or the invention of heavier-than-air flight. In keeping with this promise, we recently contracted with DARPA's new Biological Technologies Office under their Living Foundries 1,000 Molecules program. This program is developing new capabilities that will enable biomanufacturing of known or novel chemicals on demand and at scale. As few as 3 years ago, entire companies in this arena were founded to develop a single chemical product. With the support of DARPA, we at Zymergen are pushing the technology to develop new biosynthetic pathways for over 300 specific chemicals of interest. We are targeting an overall 20-fold cost reduction in new product development. Further, our team of biologists, engineers, and material scientists are choosing these chemicals to form the basis for new materials. These materials are expected to have novel properties in categories as wide-ranging as thermal-stable plastics, marine adhesives, and antiseptic battlefield dressings. While the potential application of each new material generates considerable interest, what excites me and my colleagues at Zymergen most is the creation of a cutting-edge technological platform designed to accelerate innovation in new materials, an area where innovation has slowed, and importantly, an area historically completely unrelated to biotechnology. This is but an example of the myriad ways biotechnology can impact the U.S. economy and improve society. I am pleased this hearing presents an opportunity to engage in dialogue about ways we can work together to realize the potential of this industry. Thank you.
I thank the witnesses for their testimony. Now I recognize myself for questions for 5 minutes. Dr. Shetty, you've testified that one of the potential barriers to biomanufacturing is public acceptance of these products and sometimes concerns that come up. Can you discuss that a little, both the concerns and how to address them? And I really would invite all of you, I think you've all addressed that a little bit, but how we can best proceed in this and not, and address some of the more alarming reactions in a informed and scientific way.
Absolutely. Thank you for the question, Chairwoman. Thank you for the question, Chairwoman Comstock. It's interesting. To date, biotechnology has largely been behind the scenes, right? People are not always aware that the foods they eat, the medicines they take are made with the products of biotechnology. However, I think we're seeing a shift. As the technologies are improving, more and more consumer-facing products are coming out onto the marketplace. So Ginkgo's Cultured Rose is an example. I also alluded to others in my testimony. So animal-free versions of milk, of meat, and there are companies making spider silk using yeast and spinning that into jackets. So a Japanese company named Spiber is partnering with North Face to bring out a spider silk jacket that's currently touring Japan. So I think what you're going to see over the next few years is that biotechnology is going to be interacting more and more directly with the consumer, and this is going to drive a shift in attitudes naturally. And with that shift in attitudes, you're going to see a greater public acceptance of these kind of technologies. That being said, we continue to have a responsibility to ensure transparency in our sector. So, there are obviously a lot of concerns around, what does the supply chain look like for the products I buy? Should I be seeing certain labels on my foods or the products I buy? I think really a lot of those conversations really stem for a desire for information, a desire for knowledge. And what I'm excited about actually is that through biotechnology, we can actually increase the transparency of our supply chains. If my yeast are growing these products in a fermenter in the middle of Iowa, it's a lot easier for me to understand where exactly the products I'm buying at the grocery store come from. And so, in short, I would suggest that we're seeing a transformation happen as the tools get better and better. More and more consumer-facing products are going to be coming out into the market and drive a change in attitudes.
If I could just chime in
Yes.
quickly, to be the devil's advocate, I think in the past, and I agree with much of what you said, Dr. Shetty, I would argue that academic scientists who I'm familiar with we have not done a great job in communicating to the public what it is we do and why it is so vitally important. I think that's changing now, but a lot of what you see in the newspapers are sort of peer-based, sort of newsworthy items. You never hear about the success stories that are also going on, and actually are much more abundant. So I think we just need to be sensitive to the public, in large part being uninformed properly to what it is we do. And we're all, myself as well as companies, until recently have not really dealt with that. In my view, we can do a better job of showing the great things that this powerful set of technologies can in fact do.
Okay. Well, thank you very much, and I will now yield to Mr. Lipinski for 5 minutes.
Thank you. It's been really very interesting and learned a lot here, although I still can't say I exactly know what you're doing. But I want to throw a more general question out there and have everyone tell me what you think can be done, because I know that Dr. Shetty had talked about UK and China have well-coordinated national programs in synthetic biology. So I want to start with Dr. Shetty, and you talked a little bit about this, but what do you think along those same lines that we should be doing? The big thing here is you're here to tell us what we can do to be helpful in moving things forward, and we have the best benefits for our society here. So Dr. Shetty, what would anything else you would recommend? I know you talked about a few things.
Yeah, so I think one of the things we need to appreciate is that biotechnology today is not just about health and medicine, right? And I think this hearing is testimony to that. But biotechnology in the future is going to have major impacts in many other areas besides health and medicine, including manufacturing, agriculture, and energy. So what I think needs to happen is that there needs to be a national recognition of that importance, and we need to push forward a more organized national funding program in this area. And so HR 591 is a step in this direction, and I would encourage you to garner bipartisan support for this bill and push it forward.
Thank you. Let's start with Dr. Serber. Do you have anything? So what are other countries doing? What can we do that we're not doing?
A couple of things come to mind. So amongst them is a coordinated roadmap. The efforts in the United States are fragmented. The support for this growing industry, it doesn't have a clear home base for lobbying, for support, for garnering the kind of widespread development of the tools that we are going to require to push forward the sector. I mentioned a couple of times that we're receiving DARPA support, and I believe that today in the field of synthetic biology, DARPA has far and away provided more support than any other federal agency for this enterprise. I think it amounts to roughly 60% of the dollars spent by the federal government in 2015 to this new field. DARPA is a very small agency, and they can't go it alone, and their focus is on creating and preventing strategic surprise, so their application space is understandably focused. I'm looking forward to other agencies using that as inspiration to build support base funding for additional research, both in academia and translational research. I'm looking for educational programs to help give the cross-disciplinary familiarity, because to succeed in this field requires expertise not in the silos of biology or chemistry or physics or computer science, but rather cross-trained individuals who are equally versed in aspects of all of the above to really push the field forward.
Thank you. Anyone else who wants to go? Dr. Evans?
So I think one of the things that you can see looking over the history of this technology space, the U.S., through some very aggressive, risky, early technology investments, pioneered the field. I think when people are rewriting or writing the history, looking back from 100 years or so, they will see that these early federal efforts pioneered the field nationally and led globally with the idea that engineering biology could become that next revolution on the scale of technology development along the first industrial revolution. However, as Dr. Serber was showing, there isn't now in the U.S. a coordinated framework of either the research or of how it can be effectively moved into market acceptance. And so when you look at some of the things that the other countries are doing, they are attempting to make sure that industry and academics are being mushed together to an extent. So the centers that are being funded, particularly in the UK, require some joint industry, government, and academic input. And I continue to point to SINBRC as a very good example of that domestically. But all of this will have the same challenge that the U.S. biotechnology industry had in the mid-1980s when we were trying to apply genetic engineering techniques to recombinant bacteria for release into the environment for bioremediation and other things that are going to be logical outcomes of all the biome research. So we're going to have paper after paper that says, wouldn't this be cool or important if we could do this in engineering a phytobiome? But there's not going to be a regulatory path to get an engineered prokaryotic organism out into the environment, because we just haven't dealt with those questions. They were brought up, stopped, and dropped. And so coordinating that kind of federal research that helps build extramural research centers that might be needed to deal with questions around release will be very important to realize the broad application of some of the engineering technologies that require deliberate release outside of contained fermentation.
Thank you, and I'm over time here, so I think I'm going to have to yield back. Thank you.
Okay, thank you. And I now recognize Mr. Molinar for 5 minutes.
Thank you, Madam Chair, and I want to thank all of you for being with us here today and for your testimony. And I wanted to follow up on some of the things that have been discussed. One is in the area of the coordinated research. You mentioned both long-term research, market acceptance, coordinated roadmap, strategic plan. It seems like those themes keep coming up. And then there's also the coordinated framework for the regulation of biotechnology that hasn't been updated since 1992. And I'm assuming the memo, the White House memo, was instructing that that would be updated. Is that correct? I've heard different agencies mention, the EPA, USDA, FDA. Who is the point on that? Is there one agency that's the convener in that? That's Dr. Max.
The Office of Science and Technology Policy is working with all 3 agencies because all three agencies regulate products that are biotechnology products. One of the challenges I think my colleagues have referred to is there are bioinnovations that, that straddle agencies that seem to belong partly in the domain of the USDA and partly in the domain of the FDA. And the EPA, for that matter. There are examples where all three agencies might be involved. So I think what's really promising about this is there will be an opportunity to not only to update, but also to clarify the roles of the agency. Who is the lead agency when a company wants to have discussions? So all three and the Office of Science and Technology Policy working together.
And are you all giving input to that process? Do you feel like you're at the table discussing that with them?
I know there was a recent request for information. Several companies did submit information. There will be another chance, a couple of more, I think there are 2 more public meetings scheduled. The first one was held on September 30th, I believe, at the FDA. There'll be 2 other meetings scheduled around the country starting in January, I believe. There'll be plenty of opportunity.
Okay, and then what's the timing on, would it be a new rule, a new regulation, a framework that comes out, and then there'd be a public comment period after that framework comes out?
I can't speak to the definite product of the expected products. I do know that in addition to the work of the agencies, there will, around the coordinated framework itself, there will be there's an expectation, as outlined in the memo, for a long-term strategic plan to be delivered in a fairly short time frame by the agencies. So they have a couple of jobs to do. But I do believe that there will be open comment on any product that comes out.
Okay, any others have any comment on that?
Many of the partner companies we work with who are already engaged in large-scale manufacturing are stimulated to become involved in this process by the writing is on the wall for them, that if they don't embrace the new technology, their competitiveness with the products that they make will be eclipsed by others who have. So this framework is really required to maintain the United States lead in the manufacturing of many of these goods, without which we will be stuck manufacturing products using 1980s, 1990s technologies, and others will be employing the more advanced technologies and have better economics around manufacturing.
And is the framework the same as the strategic plan, or is that totally different?
These are 2 different.
Okay.
Yeah.
And then who is driving the strategic plan?
I believe from the memo, the effort is the administration with the Office of Science and Technology Policy in concert with the 3 regulatory agencies. But I believe OSTP is working with the agencies. I don't want to say they're running it, but they're coordinating it.
Okay. Dr. Evans, did you have a comment on that?
Not on the last question. I was on your question before. You know, the thing that is important is the predictability. That is what is important for us in, say, the ag industry, where our development timelines are a decade. And so when you have policy shifts, or in particularly when you have policy frameworks that don't have a strong science base, so that you can bring data to the decision to try to move and have an informed and data-driven process, that's where things get increasingly challenging for us to make investment decisions that are reliable and robust.
Okay, thank you, Madam Chair, and thank you for your insights.
I now recognize Congressman Abraham.
Thank you, Madam Chairman. Dr. Dickman, let's go back to you for just a second. You referenced the MAGE and the CAGE, the multiplex automated genomic engineering and the computer-aided. On your research, it's certainly my belief, and I think the belief of many of us, that world security is the food scarcity or having food security for an underdeveloped nation. And I think one of the criteria for being an underdeveloped nation is that you simply can't provide enough food for your people. So, in my opinion, this is another piece of the pie that we fight world terrorism with, that we're able to feed the people that can actually do some good and do some good things. So I guess the question is, how far in your crystal ball are we away from really getting there to some of these underdeveloped nations of these technologies where you can potentially grow wheat in the middle of the desert, or you can increase yields by 5 to 10%. Where are we in the timeline there?
Well, it depends if the glass is half full or the half empty.
Right.
Certainly, there are a lot of positive progresses that are being made in developing countries, but it's a complex, it's not a cop-out, but it's a complex issue. There's a lot of politics involved, and while the growers generally support these kinds of plans and routes to food production, they're often hampered by politicians and people with other interests. So the hurdles to overcome, depending on the country you're talking to, are considerable. But I might add, but it can be done. I mean, bananas, to use something I know a little bit about, are now in human field trials, actually in Iowa, with the hope that the hoops have gone through sufficiently to put them out in a humanitarian effort in Africa in another year. That's bananas. You know, the rice, golden A rice, which they were making vitamin A in, preventing blindness in children, has run into lots of, which was been heavily advertised, and it's sort of the poster child for transgenic crop plants, has slowed down considerably due to regulatory hurdles. So to answer your question, to ever be a viable economy with an assortment of crop plants, we're probably talking 10 years as well, I would say, realistically.
Okay, thank you. That's a good enough ballpark. At least we've got something we can, maybe put our toe in the water in, so to speak. Dr. Sherburne, and I'll go to Dr. Evans. Is there anybody on the panel that wants to answer this? You mentioned the Swiss cheese, I guess, effect of our regulatory process here in the States, but you also mentioned that we need to accelerate innovation, and those two are pretty much diametrically opposed, but anytime we regulate, we slow down the process tremendously. So the balance, I'll flip to the health side for just a minute with the CRISPR technology and the Cas9. We have the potential and the ability, I think, even now to cure single mutations, single gene mutations. But again, we have countries that are abusing this to the point of trying to manufacture the perfect child or the perfect person. Where is the middle ground here? Where do we start, I guess, is a question of how we can accelerate innovation, but at the same time, make sure that this wonderful technology doesn't fall into the hands of some nefarious people.
The quick answer from and really it's just from my point of view, is the place to start is in simpler systems. The mammalian application of these technologies is more complicated when it comes to the ethical and legal considerations. The application, the technology actually began as a natural phenomenon in bacterium, and it has been applied across the animal kingdom in very short order given its power. We at Zymergen apply those sorts of technologies in the application of microbes like bacteria from which they were originally found for the purposes of improving them in the biocatalysis that they are used in large-scale fermentation. This makes for us, from our perspective, a nice test bed for assessing the suitability of the technology in a regime that certainly has oversight. I'm not implying for a moment it doesn't, but doesn't raise as many issues as other applications have. And as I think we learn more about the technology and its applications and grow more comfortable with it in this sector, it will be much easier and more natural to move it and expand it into other sectors, which will include human health.
Okay. All right, thank you. I'm out of time, Madam Chair.
I would now recognize Mr. Westerman for 5 minutes.
Thank you, Madam Chair, and thank you to the witnesses for being here today. I grew up in a time where I read stories about Dr. George Washington Carver and the amazing things that he did. Was said to be the man who could make something out of nothing with his research on peanuts and sweet potatoes. When I was in high school, I thought I was just getting out of school for a day, but I was very involved in the Future Farmers of America, and I got invited to a conference. It was called the Governor's Conference on Agricultural Innovation. It was hosted by then Governor Bill Clinton, and the special guest was Norman Borlaug. So I got to be a member of that panel. I'm not sure how that happened. If I'd known the significance of it at the time, I might have listened a little bit closer. But you know, there was a time when people who did this research and came up with all these great ideas were given Nobel Prizes. There were departments at colleges named after them. They won all kinds of awards and were viewed as heroes. Yet today, if you fast forward, as a member of Congress, I get a lot of constituent feedback in opposition to GMOs or any kind of biological research. I did also, I attended forestry school, and the time I was there, it was during the, a lot of the genome, human genome research. My undergraduate degree was in biological and agricultural engineering, so I've kind of followed this for a while. But at the time the human genome was being mapped, the genome of the pine tree was not, or it was being worked on, but it was about 7 times more complicated than the human genome. And I believe in 2014 they finally mapped or sequenced the pine tree genome with about 23 billion pairs to it. And I know that when you talk about biofuels, if you look at pine trees and you look at the amount of lignin in the tree versus cellulose, you could engineer a tree to make a lot more lignin, which would create more biofuels, or you could engineer it to make more cellulose, which would be better in paper. So there are a lot of benefits to this, but also there seems to be a lot of pushback. Dr. Dickman, do you believe that gene editing technology is related to crossbreeding or hybridization techniques that have been used for thousands of years, or is it something totally new that we should be afraid of?
You're properly managed. I'll learn one of these days. I think, again, as was stated previously, the gene editing technology is a much more serious issue in the biomedical field, because you're talking about generating transgenic people, and there's lots of ethical issues. But in terms of plants, they've been mixing, naturally mixing populations for, as you said, thousands of years, and naturally, for the most part, selecting traits of interest. What CRISPR and genome editing in general can confer to the plant field is a much more significant leap of time to get to the desired product, much more power, experimental power. So they're basically under a similar umbrella, but have different rates of progress. That is one reason why CRISPR and genome editing is so exciting, because the potential to create variation in terms of breeding practices is virtually unlimited and much more rapid and much more informed as to the breeding population. So it confers a number of advantages. Again, it comes back to the public understanding what exactly this is and how it works and why it's beneficial, as opposed to just being something with DNA related and more concern that really should be alleviated.
I know even from the forestry side, there was concerns about Franken trees. You're going to
Right.
plant these trees, and they would take over the
Monsters
landscape.
that ate Cleveland.
Right.
It hasn't happened yet.
But most of these genetically modified organisms, they require more of a specific environment to survive, and in the natural world, they can't propagate themselves as well. It's my understanding of that.
Well, that's true. There's a lot of microbes out there. I mean, as I was rushing to say in the phytobiome work, it's now become clear that these microorganisms confer a number of different traits to the host plant that they're residing in. And if you remove those microorganisms, you lose the trait, you lose tolerance, you lose disease resistance. So if we can understand the microbiome in plants or in any other, or even in the human gut where it's being done quite extensively, that gives us another avenue of approach to try to generate the types of things we need to better the world.
And Dr. Evans, what are the environmental safety and health impacts of genetically engineered plants and animals?
I think that's a great question. Those are things that have been well covered by the history of the coordinated framework in bringing products through from initial registrations in 1995, 1996 on with the original BT crops. So both nationally and internationally. These products have had a large degree of oversight. There have been hundreds of studies conducted by third parties, so independent researchers. Of course, companies have to provide data. Even the universities that are attempting to move some of these products as they try to get into field trials have to provide safety and environmental data. So I think the concept of what is there, from the large company perspective, we don't see major gaps where we could just try to drive something unregulated through the system. We have a lot of desire to be able to want the public to have confidence in these products because they're going to consume them. And so the thing we still need, though, is, after 20 years, the regulatory burden, the familiarity with the products, and the technologies don't appear to be decreasing the submission packages or time. And so things just keep getting added and added, and they do not appear always to have a strong scientific base. And so I think the federal government can help provide some research in some of the questions and independent research by federal land-grant universities and such that could help move that question down the road, because we aren't going to be able to feed the population of the planet, as has already been discussed, by simply applying and hoping for the next incremental increase in a breeding approach. There are things that need to be brought to bear in this time frame to 2050 that require novel solutions.
Thank you. Thank you, Madam Chair, for your indulgence.
Thank you. And I now recognize Mr. Tonko for 5 minutes.
Thank you, Madam Chair, and welcome to the panelists. As a nation, we're woefully underproducing scientists and engineers. In order to remain a competitive global economic power in the 21st century, I believe that we as a nation need to place a strong focus on STEM education. I fear that without an increased commitment to STEM education, American students will not be represented in the STEM fields, and American workers will be unable to compete for jobs or grow careers in the enhancing STEM industries that exist. It seems that this is the case in this area as well. In fact, Dr. Sieber's testimony mentions the need for employees with expertise in multiple relevant domains. So, to any of our panelists, my question would be, would you please discuss the skills that are necessary, essential for emerging interdisciplinary fields like the field that we're discussing here today? Anyone?
I'll start, but I think other members of the panel have something to say about it. It's worth highlighting that a panel like this is composed of people who've spent at least a quarter century in school apiece, getting the skills required to reach a level of just pure competence in the field. And it's especially difficult, given the long time horizons of the educational program, to stay current in a rapidly evolving system. And having federal support to be nimble and flexible around that, to change the educational programs and support as the technology improves, is absolutely critical. I found myself recently in conversations with faculty at UC Berkeley discussing new master's programs that they want to install with an eye towards training staff for jobs in businesses and companies like that of Zymergen, the company that I founded with 2 others, which certainly involves a lot of biology, but also more automation, robotics, computation, computer science than you would think. And I'm finding that there are certain educational programs across the United States when I go higher that are particularly adept at cross-training graduate students and undergraduates for a future in this career which will be intrinsically cross-disciplinary. It is no longer sufficient to get ahead in a technical field to be an utter specialist in one area, at least by my estimation.
Mm-hmm. Thank you. I believe, Dr. Evans, you were going to say something?
Well, I think that if you look at what we need and the students that are interesting to us, one of the places that I get a lot of encouragement is looking at something like iGEM, the program that's focused at not only the college level, but there are high school teams competing in iGEM now, and a number of them develop projects that are related to agriculture the environment. They're very sensitive to detection and remediation. And so what you have in common there is a need to understand questions and to be able to inform ways of thinking about questions that are often not just, say, one gene at a time anymore, or even one question at a time. If you start thinking about the interaction of these microbiomes, these would be multiple microbes interacting with a plant that might be interacting with an insect. So everything about it is interaction-based. And so scientific skills that can help students begin to comprehend interactions. But those interactions also have an important metaphor, which is interaction with the community at large. Just because we can do something, people need to ask the question, should we? Or how should we? And those interactions of science with their technology at the bench, being able to go have a conversation with someone who is in another department in the school with no science background at all, those skills are very unique and quite lacking. And so we need to be able to integrate a good sense of science across a number of disciplines, but the ability to think and ask questions, at least understand or comprehend questions, that there could be policy or health or ILSI implications is important.
Right. Dr. Maxson, I think you had a comment you want to share?
Yes, thank you. Sorry, I have a bit of a cold, and I'm making sure I can get through this. A couple of thoughts come to mind. In the United States, approximately 50,000 people per year receive doctorates. More than half of those people, depends on the field, of course, but more than half of those people don't end up in tenure-track academic positions. You're looking at a table here with a bunch of people who got academic training through the apprenticeship model that gives us our PhDs. But what we are not trained to do is understand the skill sets, I think, to underscore the points made by my colleagues, to work in industry, to manage a budget, to understand how to write a business plan. These are not things we learn in the system. And so on the level of graduate education, I would say that the United States should work a little harder to broaden the exposure of graduate students to the kinds of skills they're going to need depending on what field they're going to end up in, whether it's going to be science journalism or academic research or medical research or plant research at a company. It doesn't matter. I think we can do a better job at that. At the undergraduate level, a couple of thoughts occur to me there too. One, most importantly, I think we see the best outcomes in developing scientists and engineers when we give them immersion opportunities, not just canned lab experiments to do that thousands of students before us have done, but actual research experiences where we're the first people to ever actually do an experiment in an undergrad environment. I know that's hard to do, and I know it's expensive, but there are people who are doing it, and I think it's a very good trend in the right direction. And lastly, community colleges. I know some of the national labs, ours included, are working very hard to establish relationships with community colleges to put into the curricula critical, inspirational pieces for understanding how to engineer biology. So I think there's a lot of work we can do.
Thank you very much. I think you've all cited a need for investment, and I endorse that. Madam Chair, I don't know if Dr. Shetty had any comment. It looked like you wanted to share some thoughts.
Sure, you're welcome to.
Yeah, the one comment I want to add to my fellow panelists is that STEM education needs to start early. Doesn't begin at the undergraduate level. I myself had the benefit of doing a research experience at my local university as a high school student, and those early exposures to STEM education is critically important to fostering the scientists of today, particularly when you're talking about young women, right? There are a lot of, the balance of genders between men and women in science and math fields is very skewed in a certain direction. And so we're not tapping into the full potential workforce with those statistics. And so as I look forward, encouraging young girls to participate in STEM fields is absolutely important. And as a mom with a young daughter, I want that for her.
Super. Madam Chair, thank you, and I yield back.
Thank you. I'm glad we got to have you mention that opportunity. I started a Young Women's Leadership Program so we could do that very thing, and my daughter is a biology major, did not get any of that from me. So, but she had a lot of great women teachers at George Mason here in her master's program. I'll now recognize Mr. LaHood for 5 minutes.
Thank you, Madam Chair, and I want to thank the witnesses for being here and for your testimony and all the work that you do. Dr. Shetty, question for you. There's been discussion about how the United States is losing our competitive edge with China and the United Kingdom when it comes to synthetic biology. And I guess trying to understand the reasons for that and why we're falling behind and what steps we need to take to maintain our competitive advantage in biotechnology.
Yes, thank you for the question. So I think probably the best example of interest in the worldwide interest in this area is the International Genetically Engineered Machines Competition. This is an undergraduate competition in synthetic biology where teams from universities design and build genetically engineered machines, organisms. And for the past few years, most years the winner is not from the United States, right? It's coming from Europe, coming from China, coming from overseas. And so I think this is a reflection of what is to come if we don't make domestic investments in this area. And so I think part of the problem or part of what needs to happen in this country is that we need an organized program of investments, right? No one piece is enough because there's a lot of synergy to be had between having the agencies understand and coordinate their research efforts both on the basic R&D side, but also on the translation into industry through SBIR programs. And then finally, as I alluded to in my opening remarks, the U.S. government serving as a customer for biotechnology products as these nascent industries are getting going. And so we need a coordinated multi-pronged strategy, and that coordinated strategy has been pushed forward by the United Kingdom, by China, by other countries in the EU, but so far we have not done the same here in the U.S.
In the competition that you referenced, when did that change occur where the United States hasn't been the winner? Was that recently or 5 years? How long ago?
The competition started in about 2004. I would say by 2005, 2006, there started to be, the winners of the competition started to become schools from outside of the U.S. rather than within the U.S., even though this field largely has its original roots in the United States. I was there. I was part of it.
Gotcha. Dr. Maxson, as a follow-up, my home state of Illinois has a large and diverse bioscience industry with over 78,000 jobs and 3,400 businesses that contribute to the state's economy as it relates to bioscience. I know you were the author of the National Bioeconomy Blueprint in 2012 that outlines steps that federal agencies should take to drive the bioeconomy in the United States. I know we referenced that a little bit earlier, but what's the status of those recommendations in that report?
Thank you for that opportunity to talk about what's happened since the release of that policy document. I think the recent memo on July 2nd from the White House talking about taking a look at the regulatory framework, the coordinated framework, is a direct reflection of one of the 5 strategic objectives of the National Bioeconomy Blueprint. So I would say in that regard, right at the top of the list is taking a look at the coordinated framework. Workforce development was another. You've heard some ideas of how we might be able to jumpstart the system and get a few more chemical processing engineers, that kind of thing. Still needs some work to be done there. Public-private partnerships for biosciences. I think what could be done there, and there are some efforts underway right now, I believe, funded by the NSF, to identify pre-competitive research challenges that industry shares that might actually benefit from public-private partnership with both government and company investment. So those are 3 right away. A couple more. Strategic research investments. That was the #1 objective in the National Bioeconomy Blueprint. I think you've heard most of the people, if not all the people on the panel, say the same thing. We could do a better job here. And one of the reasons, to answer your last question, to address your last question about why are we falling behind in synthetic biology specifically, I look at this as another example of technology. Nanotechnology is an example. Emerging technologies. Technology in general sort of falls between the cracks in the federal agencies. And so I think the idea of a coordinated, federally coordinated strategic approach to lift the technology is where I think some opportunity still remains.
Thank you. Madam Chair, if I could ask one last question, Dr. Shetty. Okay. Thank you. Dr. Shetty, one of your company's projects funded by DOE ARPA program supports R&D to capture natural gas flared by shale. Can you describe how your company is using that biotechnology to conduct this work, and what have those outcomes been thus far?
Thank you for the question. So there's an interesting transition that's happened in recent years in this country, which is that on a per carbon basis, carbon derived from methane, natural gas, methanol is cheaper than carbon derived from other sources, say sugar. And so there's a growing interest both within our company and others in using these as feedstocks for bioproduction of various chemicals and fuels. And so we have initially had DOE ARPA-E funding in this area to develop some nascent technologies, and we've since partnered that work with a commercial partner and are taking it forward. Now, unfortunately, because it's partnered, I'm under some confidentiality restrictions, and so I'm not able to speak to the details of that program. But suffice it to say, this is an area of interest both for us, ourselves and others, and it's a potential new frontier when it comes to bioproduction of these types of fuels.
Thank you, and I now recognize Mr. Hultgren.
Thank you, Madam Chair. Thank you all so much for being here for this important discussion. I do believe this is an important hearing, and as technology continues to evolve and new opportunities materialize, it's increasingly necessary that we keep our regulatory structure up to date while developing biotechnology in the most ethical way possible. This means coordination and communication between our researchers, their institutions, our government, and also among government agencies. Dr. Maxson, I wonder if I could address my first couple questions to you. You mentioned the Human Genome Project in your testimony, which for me has been an excellent example of the unique capabilities of the Department of Energy to bring to the table in computing, among other things. DOE basically had to start the project and prove the concept before NIH was able to take this up as a serious, cost-effective endeavor. How has the Human Genome Project benefited the non-human health biotech sectors? And also, is there a similar systematic sequencing project needed for agriculture or naturally occurring chemicals as well?
Thank you for your question. I apologize, I have a bit of a cold today. Your first question, Human Genome Project, how has it benefited the non-biotechnology? I assume you mean the non-biomedical world.
Yes, I'm sorry.
No, thank you for clarification. I think one thing that the Human Genome Sequencing Project did was democratize DNA sequencing. So laboratories everywhere, whether you're studying viruses or the plant microbiome, whatever it is, people now can sequence DNA very quickly as a consequence of the Human Genome Project, the Genome Sequencing Project. So I think that, in fact, I don't think it's overestimating it to say all of biology has benefited in that way. Anything that has DNA, if you can sequence DNA quickly and cheaply and in a democratic fashion, everything has benefited. So I think the magnitude of that can't be underscored. If you could remind me of your second question.
Yeah, second question was, as far as agriculture or other naturally occurring chemicals, is there a similar systematic sequencing project that we need or that we should focus?
A similar systematic sequencing project. Wow. I am not in a great position to answer that question. I think I would defer that to my agricultural colleagues.
Does anybody else have
Yeah.
Yes.
Yeah, there's actually quite a bit being done in agricultural sequencing, if you will. In fact, NSF has a plant genome program that is actually very well funded to hear, and has been ongoing for a number of years. There's also a microbial genome sequencing program that just finished. So, and also independently, now that it's gotten so relatively inexpensive and available and doable in a rapid fashion, there's a great many agricultural-related genome sequencing projects going on now. Another area to be marketable in is bioinformatics and computation, because back when I was a student, you know, you cloned a gene with your thesis. Now you go home, it's 25,000 genes, and you have to figure out what to deal with it. So there's a massive amount of data handling, but it is being done to the U.S.'s credit in support of those kinds of projects.
That's great, thank you. I'm going to go back to Dr. Maxon, if that's all right. From your time at OSTP and now at the lab, surely you've seen the difficulties of getting agencies to work together, especially in getting them to leverage one another's resources, tools, and human expertise. It sounds to me like there is great potential if agencies would work more closely together in this space. For instance, if ag aggressively leveraged the synthetic biology and genomic capabilities of the national labs. I wonder, will this work, and what do you suggest? How do we, from your experience, how do we best work together?
Thank you for that question. I'm tremendously optimistic about this. I do think there's an incredible opportunity here. The potential is amazing. I was heartened to see the President's Council of Advisors on Science and Technology in December of 2012 released a report called Agricultural Preparedness. In that report, they recommended that the USDA work with the DOE and the NSF to set up new innovation ecosystem hubs for agriculture. I think an idea like that, where the DOE, that knows how to set up innovation hubs, working with USDA, could go a long way with NSF in making something like this happen. I was also heartened to see, not long after that report, that the USDA, in its budget, requested funds for a biomanufacturing institute. So I think we're very close, and I'm very optimistic. So I think it will work. It just might take a little bit more time.
Great. My time has almost expired. I will yield back my last 7 seconds. Thank you.
And I will now recognize Mr. Weber for 5 minutes.
Thank you, Madam Chair. Let the record show I have 5 minutes and 7 seconds. Thank you for the opportunity to be here and participate. And Dr. Dickman, this question is for you. This past year, public researchers involved in communicating the science of biotechnology and its impacts have actually been targeted, both professionally and personally. I'm sure you're aware of that. Doctor, as a public scientist, can you give me more background kind of into the current academic feeling on this public outreach? Y'all are getting targeted. A lot of some stuff's been aimed at you. What's the feeling amongst your peers?
Well, a number of things, disappointment and things of that nature. When you see a greenhouse that's been destroyed by stones, for example, with all kinds of messages written on them, it's a bit disconcerting. But in terms of people's research, I don't think that has impacted it. I mean, people are still doing what they plan to do and continue to do and get funded to do. So while it's an unfortunate circumstance, I really don't think it's really had a strong impact on people's ability to do work, with the exception that there is some material things that have been destroyed and needed to be replaced. It's actually not too bad now, I would say.
Not too bad. You actually said, I believe in your discussion with the chairwoman, that you felt like you all needed to do a better job of showing capabilities and I guess educating the public.
Very much so.
Has that been progressing?
Well, I do it on a sort of a grassroots level. We don't have any organized framework with which to do this. I think we do. We do need that, whether it be from academic or companies.
Have you done the genomic sequencing on that grass? You said grassroots level.
That's actually in the queue for other reasons.
All right, so you're doing it at the basic level is what you're saying.
Well, there's a number of turf breeders who work strictly on golf course turf you might want to talk with.
There's a shock, huh? Well, thank you for that. And Dr. Evans, as you know, Dow Chemical has a lot of industry in my district there in Texas. In fact, I was going to tell the gentleman from Illinois, he left before I could, that there was 78,000 jobs associated with this. In Texas, there's 81,000 jobs. So things are bigger in Texas. So, but Dr. Evans, you also mentioned in the 3 C's of national needs, both continuing to support national scientific funding agencies and convening forums of discussion for that public engagement or outreach that Dr. Dickman and I were just discussing. With limited resources in public research, what role do companies, for example, like Dow Chemical play in promoting that scientific interest?
I think one of the ways we have been involved last year with the help of the NSF, the Woodrow Wilson Institute, there was a convening of companies, regulatory bodies, and non-governmental entities that had interest in the environmental release of microorganisms, whether they be algal strains that might do chemical production, or concepts that synthetic biology might want to bring into the environment. That group at least published recommendations for where things could go, with some of those recommendations being specific federal funding. Now, companies, I do think, need to be able to know where to direct their research, and their research needs to be aimed at their product technology space and legitimate questions around that product area. But there are some things that just are big enough in scope, or they are fundamental questions of biology or biotechnology that are more properly addressed in integrative lab studies from multi-university settings, or they might be appropriate to be something that would be the outcome of a national lab and a focused program. But industry could then, even in that scenario, be an appropriate partner. I think the thing from the public perspective is we need to make sure that the public can see transparently where those contributions are being made and impact.
That's a good point. You know, in your discussion with the gentleman from New York, Representative Tonko, I think you said just because we can do something, you should ask the question, should we do something? And if the public perceives that a company is getting involved, is that a conflict of interest? I think you were the one who said, let me quote you earlier, you said that we needed more feed, fuel, fiber, and food more by 2050 than in the last 10,000 years. That's an astounding fact. And with limited resources available, I think if the public knew, you know, what was at stake here, that they might not be so suspicious. But I appreciate your testimony, and Madam Chair, your indulgence, and I yield back.
Thank you. And I think by agreement, Mr. Westerman has a few more questions, and I'm going to let him have the chair because I'm going to have to depart. But I want to thank the witnesses very much for a very interesting and insightful hearing, and appreciate all the great research you're doing. And I'm glad we have two women here too, so thank you. And we certainly appreciate the men, but thank you for your comments, Dr. Shetty, and we'll keep those in mind going forward too. Thank you.
I guess that's one way to get to ask another question. Dr. Maxson, you mentioned this briefly in some remarks, but talking about nanoscience, and I was able to tour the Institute for Nanoscience and Engineering Technology at my alma mater, the University of Arkansas. It was very exciting to me, the possibilities there. So I was wondering if the panel could address the opportunities in nanoscience as it relates to biotechnology, and is this an area that needs more research funding?
I'm not an expert in nanotechnology, but I'll kick this off and then allow my colleagues to respond. I know that nanotechnology intersects with biotechnology in some of the high-level treatments that are being done now to target certain therapeutics to certain parts of the very specific parts of the body. I know that nanotechnology is used in the process of doing some diagnostic kinds of analyses, again, in the biomedical space. Like I said, I'm not an expert. I don't know much about how nanotechnology intersects with the non-biomedical fields. It would be interesting to hear from my colleagues whether there are any.
There was, in fact, a small NSF industry and university consortium that was established at the University of Illinois to try to bring together, it was established at a former nanotechnology center. Well, it still is a nanotechnology center, but they brought in industry that was involved in agriculture, some other industry that was involved in food and diagnostics and medicine to try to come in and bring products to market rapidly that could be based on nanotechnology. I think if you just step way back, I'm not a physicist, but the thing that nanotechnology did to material sciences is help re-envision what was possible. We thought we knew what was possible with our understanding of the physics and of the performance of materials at a certain scale. Nanotechnology changed that, and remarkable products and concepts came out of that. I think engineering biology is doing the same thing to biology. We had a framework of what was possible that was rocked with the development of recombinant DNA technology in the early 80s. Insulin came very quickly after that, a Nobel Prize. Now we have high school students that could do the same level of engineering that formed early products. And so this is re-engineering, reimagining what is even possible using biology for what it's very good at, making things, making nanostructured things. Biology makes wonderfully complicated nanostructured materials in things besides carbon. And so how does it do that? And how could technology be brought to bear to do that? And so I think it's questions like that that a good, well thought out national plan for bringing students and bringing the technology to bear could follow on that metaphor of nanotechnology. And it had to bring in associated concepts of regulatory and safety all at the same time.
Dr. Maxson?
To follow on the point that Dr. Evans just made, the National Nanotechnology Initiative is a great example of a coordinated effort that gave rise to opportunity, coordinated federal research, coordinated interest in public science, the public understanding of the science. I think that that model exists that could be applied here.
Anybody else like to address the nanoscience question?
Only very briefly that biology is already the supreme source of exquisite molecular structures. Biology and the enzymes that it employs to do chemistries are there for us to make use of as we attempt to expand the chemical palette of building blocks that we can use to make new materials. So there is a definite overlap in some of the applications.
Dr. Zerber, just as a quick last question, your work in biofuels, can you describe some of the barriers that exist for bringing biofuels to market? I know there's been a lot of attempts, really not any successful attempts.
Yes, quickly. So I am currently no longer working on biofuels, but I did spend about 7 years pursuing biofuels. And the challenges that sector faces are driven by the macroeconomic forces having to do with the price of oil and the price of feedstocks for the fermentation products. It's worth highlighting that in the course of pursuing biofuels, both with private and federal funding, all the tools that this panel is making use of to drive other applications and other of the technology, a lot of that began with biofuels. The biofuels have not yet reached the economic tipping point to be competitive, but things only need to change a little bit for that to turn around, and we'll be ready when they do.
Okay. I'd like to thank the witnesses for their valuable testimony and the members for their questions. The record will remain open for 2 weeks for additional comments and written questions from members. The witnesses are excused, and this meeting is adjourned.